Now what I want to do is take you behind the scenes and show you how we're going to make the solution for our osmosis in a model cell portion of our cells and their membranes lab. After we make that solution, we're going to pour it into a model cell. So the solution is going to represent the cytoplasm of our cell. We're going to let our model cell sit in a beaker of water for about 30 minutes. And at the end of that 30 minutes, we're going to test to see what was able to pass through the membrane of our model cell and what molecules were too large to pass through the membrane of our model cell. To make our model cell, what we're going to be using are 3 macromolecules. We're going to be using spray starch to put starch into our cell. Spray starch is normally used by people to help iron their shirts. We're going to use albumin, which is a protein that's found in egg white, and we're going to use glucose, okay? We are going to put those into our fleeker here, half flask, half beaker, filled with water to create our cytoplasm. And then we're going to pour some of that solution into this semi-permeable bag. It's made of a special tubing called dialysis tubing. We're going to pour some of our solution into that bag and then put our bag into a beaker. So to begin with, I'm going to start with the spray starch. I'm going to take the spray starch and just hit my solution. I'm going to shake it up a little bit. Hit my solution with a bunch of starch. And that starch is soluble. It'll dissolve right into solution. All right, so now we have some starch in our cytoplasm. Next, I'm going to put some albumin in. There we go. Get a little protein in there. Okay. And then last, the dextrose, the glucose. Okay, so I'm going to pour a little bit of glucose into my solution. The amount doesn't really matter. There we go. Got a little glucose in there. Okay, and now I'm going to stir this up a little bit. Get it all mixed together. Okay. And after I get it mixed together, I'm going to pour it into this bag, tie off the bag, and put that bag into a beaker of water. Now that my cytoplasm is all mixed up, I'm going to take it over here. I'm going to pour it through a funnel into my dialysis tubing, filling my dialysis tubing part way up. That should be good. So there goes my cytoplasm. Take out my funnel. And now I have my dialysis tubing filled part way with cytoplasm. This dialysis tubing, like I said, is a semi-permeable membrane. I'm just going to tie this off. Okay, just going to tie this off. There we go. And I've got my dialysis bag with the cytoplasm inside of it. This is my model cell. Now what I'm going to do, I'm going to take my dialysis tubing. I'm going to make sure that I rinse the outside off in case I spilled any of the cytoplasm on the outside. Once I get it rinsed off, I'm just going to take this dialysis tubing and I'm going to put it into this beaker where I just have some water. And now I'm going to just let that bag sit in this beaker for about 30 minutes and allow some diffusion to take place. And then we're going to test with iodine, Biuret solution, and Benedict's indicator to check and see what has been able to pass out of the bag and into the beaker, right? So I'm going to take a little bit of the solution out of the beaker, put it into 3 different test tubes, and I'm going to do an iodine test, I'm going to do a Biuret test, and I'm going to do a Benedict's test. And what I might do with the iodine, instead of putting something into a beaker to test with iodine, I might actually, after taking some out to do a Benedict's and Biuret test, I might just pour some iodine directly into the beaker, and we'll do our test for starch that way instead. So the bag has been sitting for around 30 minutes now, and I'm going to do 3 indicator tests with it. I'm going to do a Benedict's test, a Biuret test, and an iodine indicator test. I'm going to start by taking out a couple of pipettes full of the solution from my beaker, and then I'll get the Benedict's test started and do a Biuret test. So here we go. I'm going to withdraw some of this solution. put it into a test tube, get a little more. Okay, so that is test tube number 1. To that, I'm going to add, I don't know, 5-ish drops of Benedict's. And if you remember, Benedict's requires heating. So I'm going to take my test tube and I'm going to put it into a hot water bath. So I'm going to let that sit for a little while now. The second test we're going to do is check to see if the protein, the albumin protein that I put into the bag, was able to get out through the bag into the solution in the beaker. So I'm going to do a Biuret test, and this does not require heating. So I add some Biuret. I'm going to add my solution. I probably should have done this in the reverse order, but that's okay. Get it all mixed together. All right, so I'm going to add some more Biuret just to be sure. Okay. Put that back in. Lift this up. Take a look. And it has not changed that lavender color we would expect. It is still blue. Okay. Benedict's test is still going. We'll check in back on that in a little bit. The next thing I want to do is I'm going to do an iodine test. So I need to get the lid off the bottle, which it is pretty well stuck here. So we're going to take it. There we go. So we'll get that off. Take that off. Okay. Remove my dropper. And what I want to do is add the iodine to my beaker. I'm going to wait on that until the Benedict's test is done. When the Benedict's test is done, we'll do the iodine test. Checking back in on our Benedict's test, we see that the solution is turning orange from the blue that Benedict's is, which of course indicates that monosaccharides are present in our test tube, which means they were also present in the solution in the beaker, so they must have gotten out of the bag. So the last thing I want to do is I'm going to pour some iodine in to this beaker, quite a bit. And we notice that the iodine doesn't change color when it comes into contact with the solution in the beaker. I'm going to put this beaker over here, and what I'm going to do is I'm going to put my phone up on that stand, and I'm going to take a time-lapse video of the iodine in the beaker. So what we see now at the end of our Osmosis in a Model Cell lab is that the solution around our bag is all still yellow, like the color of iodine, yet inside of the bag is a deep dark blue or purple. So you will have to figure out what that means. So that concludes our Osmosis in a Model Cell lab. Hopefully you have learned a little bit about diffusion and osmosis from observing this lab. You'll need to fill out your lab handout, and we will move on to the next part of our lab.